class. My name is Tuan, and I'm currently a third-year PhD student in Llewellyn Lab at San Diego State University. And we are a group of researchers wanting to study the host pathogen interactions and innate immunity. And we're doing this by using the nematodes and their associated, naturally associated pathogens as a model to dissect these infectious disease and their biological processes. So the nematodes typically C. elegans as a model organism, and there's some other related species. They have a variety of pathogens, including virus, some fungal-like pathogens, that's called microsporidia, and they also have a variety of bacterial pathogens. So in our lab, we are interested in the interaction between intracellular pathogens and the host cells. And we recently discovered a new bacterial species that is a pathogen, a natural pathogen for the nematode Ochias tubuli. And this bacterial species is intracellular. So now, as you may know, bacteria pathogens can be classified either as extracellular, where they stay outside of the host cells, and they, relatively speaking, they have less resources. But they can, once they can replicate, they can disseminate a little bit more easier compared to intracellular bacteria. The reason why intracellular bacteria has a hard time disseminating is because even though they're inside the host cells, which is a rich environment providing with a lot of goodies for their replication and growth, because of the lateral membrane that distinguishes the cells also represent a barrier that the bacteria has to overcome to disseminate from the originally infected cells to the neighboring cells. And we are interested in the questions of how these intracellular bacteria disseminate from one cell to the other. And most of the study in intracellular bacteria dissemination so far has been converged on three main paradigms. So for some bacterial species, they can simply stay in the cells, replicate, and using the host resource. And at some point, the host cells die and lies, and they will get out of the cell and then reinfect the neighboring cells. And this is a typical scenario for some chlamydia species. The other mechanism that bacteria can disseminate from originally infected cells to neighboring cells is that they usually hijack a host actin network to form what we usually call actin tail that can propel the bacteria through the cytoplasm, and then at some point they will form a protrusion into the neighboring cells. This is the protrusion with the bacteria in it. And this protrusion eventually engulfed by the neighboring cells and form a vesicle that containing the bacteria. And with some relatively less known mechanism, the bacteria can escape this new vacuole and then reinitiate the next round of infection. Or in certain cases, the bacteria can also secrete factors, and these factors basically break down the membrane that distinguishes the cells. And if you can imagine, basically these two cells eventually merge together and form a nucleated cells. And that is how the bacteria can disseminate to the next cells. So what we found interesting in our new bacteria is that they don't really form any of actin tail or that we have in synapse. They usually just form upon invasion. They get inside the cells and they convert from a coccobacilli form to a long filamentous form. And these filaments seem to traverse through multiple intestinal cells of the nematode. And we hypothesized that this filamentation is used as a mechanism to spread among cells. And we are currently trying to dissect what are the molecular mechanism of the filamentation process in this bacteria, and also what are the triggers, the molecules inside the host cells that trigger the bacteria to undergo the morphology change from the coccobacilli form to the filamentous form for spreading. So that is the current work we are doing in our lab, and hopefully I can have some new result to tell you guys soon. In the meantime, thank you for listening and hope you enjoy the other video clips in the series. Thanks.